Yeah, so I think a lot of the folks on the call from the Dewpoint side already know that you did a postdoc with Ruth up in New York, and I guess today you'll mostly be talking about that work that you led in Ruth's lab on the self-assembly of the germ granule mRNAs. So take it away.
Okay, so thank you. Let me just share the screen. Okay. Okay, so, okay, this is good. Okay, so yeah, thank you so much for inviting me and to really share the work that was done in Ruth Lehman's lab. And I'll come to the slide in a second where I'm going to acknowledge that. So today I'll show you how mRNAs in Drosophila germ granules, upon their enrichment in these germ granules, self-assemble and they form homotypic clusters, and how this self-assembly is independent of any particular RNA sequence. As I said, and as you mentioned, the work that I'm going to show you today was really done in Ruth Lehman's lab when I was a postdoc in her lab at NYU. And I would like to acknowledge her and thank her, as well as my collaborators in the lab, as well as outside, and of course also my funding. But just to introduce, my lab is interested in how mRNAs in cells are spatially organized and what are the biological consequences of such organization. And as you might know, the organization or localization of mRNAs in cells is very important because it helps, one, establish polarized cells. As example, in this mouse embryonic fibroblast, when beta-acting mRNA is concentrated and localized to the leading edge of this motile cell, where it becomes locally translated and feeds actin monomers into a growing cytoskeleton to help this motile cell propel forward and to keep the forward motion of this migrating cell. The spatially organized mRNAs are also very important in early development. An example is Drosophila embryo. So these early embryos are completely transcriptionally quiescent, and about 30% of all mRNAs that are deposited in this embryo are somehow spatially organized. And this really is important because it helps build a body plan of this developing organism. However, this process can also go wrong, as you can imagine, and can cause disease. An example is myotonic dystrophy. It's a genetic disorder in humans that is caused by massive overexpansion, massive overexpansion of CTG repeats in the 3' UTR of this mRNA. And when this aberrant mRNA is transcribed, instead of being transported out into the cytoplasm, it accumulates in the nuclei and forms inclusions, these RNA aggregates. And these aggregates really are the one driving the disease. This is an RNA gain of function mutation and causes progressive muscle weakening and premature death, and of course, myotonic dystrophy 1. And as Ankar Jain and Ron Vale have shown, the likely cause of aggregation or assembly of this inclusion is the CTG repeats that are massively overexpanded, where the CTG repeats can base pair with neighboring DMPK mRNAs to cause these aggregates. So it's fundamentally important for a cell to really know which mRNAs it must spatially organize and which it must not, and when to organize them and where in the cell to organize them, because that can really mean the difference between normal cell growth as well as the disease. And my lab is interested in a particular type of spatial organization of mRNAs, and this is enrichment of RNAs in RNA granules, as you know very well. We know a diverse type of RNA granules, those that are forming in every cell type, such as processing bodies and stress granules. And there's more, there's those that are more special and form in particular types of cells, such as germ granules that form in germ cells, or neuronal granules that form in neurons. What is common to all of these granules is that they're all round, they're non-membrane bound, they're full of RNA binding proteins and mRNAs, and it is thought that very important post-transcriptional regulation is occurring in these granules, and that this post-transcriptional regulation is fundamentally important for specific functions that are required by the cell. For example, the germ granules are thought to regulate mRNAs that enrich in these granules to control the germ cell fate. neuronal granules are thought to promote neuronal shape and function. Processing bodies are thought to be the sites of mRNA decay, and stress granules are thought to be the sites where mRNAs accumulate during stress and are protected and translation silenced during stress. Many of these functions, especially in terms of post-transcriptional regulation and the mRNA, what happens to these mRNAs, how they arrive to these granules are still largely unknown, though much work has recently been focused on addressing some of these problems. We now understand that specific features, either sequence features or structural features in the RNA itself are required for enrichment or recruitment of these mRNAs to granules. However, it is largely unknown what happens with the mRNA once it arrives to granules. How are they organized? How are they relating to other RNAs? And how they are actually regulated in terms of post-transcriptional regulation? And some of these topics, specifically how RNAs are organized in these granules, I will talk about today. So to address these questions, we use the establishment of the germline in the Drosophila to understand this process. And we really like this model system because it first and fundamental steps in the establishment of the germline of germ cells really rely on post-transcriptional regulation, in fact, exclusively initially. And the first critical step involves localization of a subset of mRNAs to the posterior tip of this fertilized egg, where these mRNAs accumulate in germplasm. This is a specialized cytoplasm that accumulates particular proteins called germplasm proteins. And these germplasm proteins condense into small granules called germ granules, as shown in green here. And they attract mRNAs, particular type of mRNAs. And these mRNAs become regulated post-transcriptionally in these granules. They're only translated in these granules. The counterparts that are not enriched in granules are translationally repressed. They're protected from decay. And the protein products that are coded by these mRNAs then instruct the establishment of germ cells. So to understand what happens with these mRNAs and how they're organizing in these granules, we used fluorescence in situ hybridization and super resolution microscopy. We used commercially available SM fish probes. These are small DNA primers that are labeled with a single fluorophore. And we used many of them that are aligned at different parts along the mRNA. And this really was helpful because it amplified signal to noise ratio, and of course, increased our detection sensitivity. And we coupled SM fish with a structured illumination microscope. This was a particular version of this type of microscope developed by Harry Schroff at NIH, and we've been collaborating with him for many years. And this microscope provides resolution that's about twice better than a conventional confocal microscope, and really in combination with SM fish, enables us to see how mRNAs are distributed both in germplasm as well as in the somatic regions of the embryo with very high definition. So we looked at one of these mRNAs, nanos. I'm showing you here an example. And what we found was that this RNA outside of the germplasm, here in the somatic regions of the embryo, is mostly present as a single mRNA, but as soon as it enriches in granules, it forms clusters, where these clusters contain multiple mRNAs derived from nanos gene. And on average, the number of nanos mRNAs per nanos cluster is about 14 mRNAs. And as I said, we see this type of clustering in granules and single mRNAs being outside for many mRNAs that we have looked at that have a capacity to become enriched in granules. So we wondered how these mRNA clusters belonging to different genes relate to each other within the granules. And we looked at cyclin B, nanos, PGC, and GCL mRNA clusters. We labeled them with SM fish probes, here showing in magenta. And we asked how they're relating to each other, but also how they're relating to the protein content of the granule, labeled here with the core granule protein VASA, tagged with a GFP. And what we found was that they were not all homogeneously distributed. Instead, some mRNA clusters, such as those belonging to cyclin B, were located in the center of the granule. Those belonging to GCL were located more at the periphery, and nanos and PGC mRNA clusters seem to be localizing somewhere in between. we wanted to characterize this colocalization better quantitatively. And we employed two measures of colocalization to do this. In the first, we used a spot detection algorithm that measured the nanometer distance between overlapping mRNA clusters and germ granules. And in the second, we used Pearson's correlation coefficient, Costas approach. In this approach, we measured how the fluorescence intensity of two objects that are labeled with spectrally distinct dyes are correlating with each other within the overlap. And PCC Costas is a really great measurement because it's insensitive to the duration and the frequency of the overlap. So you can do these measurements even in crowded environments such as germplasm. It ranges from 1 indicating perfect colocalization to 0 indicating random colocalization. And as the distance between overlapping objects is increasing, PCC Costas is decreasing, as shown here, indicating that now, as it was the case with our measurements, mRNA clusters and granules are now colocalizing at a greater yet fixed distance relative to each other. So we used these two measures and implemented them on our mRNA clusters, and indeed we found that some mRNA clusters, such as those belonging to cyclin B, reside in the center of the granule. You can see very high PCC Costas for cyclin B and short distance, whereas those belonging to GCL clusters localize much more at the edge. You can see a smaller PCC clusters and longer distance, and nanos and PGC clusters are found somewhere in between. We then use the nanometer distances that we have obtained when we were measuring distances between individual mRNA clusters and VASA granules, and use triangulation experiments. And we've done this together with Timothée Lyonnais at NYU. And we simulated an average three-dimensional structure of the mRNA-bound germ granule. And what this experiment really showed us that indeed these clusters were occupying distinct positions within the granule. These clusters weren't mixing, and that therefore they were homotypic in nature. But what was really, really surprising was that when we looked at core granule proteins, Oscar, VASA, aubergine, and Tudor, these are the proteins that arrive at the posterior first, make the granule, and recruit all other components directly or indirectly, including mRNAs. These proteins in turn were homogeneously mixed. So you can see a lot of white overlap. So that was perplexing to us. How was it possible that within this unorganized protein environment, you get these mRNA clusters that are so highly spatially organized? And this is what we wanted to pursue. And this is the work that hasn't been published, but it's now in review and in the final process of revision, and will soon be published. So very early on, we've noted that mRNAs that can form abundant mRNA clusters, meaning they have many mRNAs per cluster, will reside in the center of the granule, as shown by this curve here. And those mRNAs that form less abundant mRNA clusters will reside at the edge. For example, cyclin B mRNAs form clusters that on average contain 30 cyclin B mRNAs, GCL, and reside in the center. GCL clusters on average contains 2.8 GCL mRNAs and reside at the edge, and nanos somewhere in between. So then we thought to deregulate or to change the expression levels of these mRNAs in the entire embryo, and to thereby change the abundance of these clusters at the posterior pole to see what happens. And we first focused on cyclin B and nanos, and we used underexpression experiments and drove down the expression of these RNAs, and these embryos now form less abundant cyclin B clusters, as you can see here, and less abundant nanos clusters, you can see at the bottom. And this really caused these clusters to move towards the periphery of the granule. And in converse experiment, we then increased the expression of GCL. And when we did that, embryos formed more abundant GCL clusters, and now these clusters were pushed towards the center of the granule. So clearly, within this unorganized protein environment, in vivo, these mRNAs have a capacity to self-organize and even determine the position, their own position of these clusters within the granules by themselves in an mRNA concentration dependent manner. This was also very intriguing to us, and we wanted to understand it a little bit better. We were intrigued by this ability of mRNAs to self-organize, and we wanted to understand how. So the two models that we thought about was that this self-organization, self-assembly could be mediated by RNA-binding proteins that are deposited on the mRNA in a sequence-dependent manner, or that mRNAs are engaging in direct base pairing through intermolecular trans-RNA-RNA interactions. And we wanted to test this. So in this model, homotypic clusters can then arise either through specific protein-protein interactions, specific RNA-RNA interactions, or some combination of the two. So the way to test this, we thought that if we had a scenario where we express two mRNAs that share features, RNA features that are required to generate the specificity for homotypic assembly, but were otherwise distinct, then these two mRNAs would populate the same granule and also condense into the same homotypic cluster. But if those features were not required to generate the specificity for homotypic assembly, then these two mRNAs would come to the same granule but form two distinct clusters. So we focused again first on nanos, and we started with the nanos 3' UTR because the Lehman, Gavis, and Wharton lab have previously shown that the specificity required for the enrichment of this RNA to the germ granules is encoded in the 3' UTR of this transcript. So we thought that maybe features that are driving homotypic assembly of nanos in granules might also be encoded in the 3' UTR. So we created chimeric mRNAs that were fused with a 3' UTR of nanos and were fused to the open reading frame of some foreign transcript ORF belonging to the far right protein. And this also accommodated binding of spectrally distinct probes and differentiation between endogenous nanos and this chimera in the embryo. And were driven by some form promoter, and they were tagged with a 5' UTR of some other gene. And when co-expressed and labeled with SM fish probes, indeed they were localizing to the same granule, but they were not forming, coming together to the same homotypic cluster. You can see there's not a lot of spatial overlap between the chimera and the endogenous gene, and even the quantification itself indicated the colocalization was low. So this experiment really indicated that the 3' UTR features, although required to bring this RNA to the granules, are actually completely dispensable for homotypic assembly of this RNA once the RNA arrives to the granule. So features are not, for this homotypic assembly, are not found in the 3' UTR. We then tested also the promoter, the 5' UTR and open reading frame, either in the combination or alone, and found that none of these features were required for homotypic assembly of nanos mRNA within granules. So this idea that these RNAs are assembling because of specific sequences, either through RNA binding proteins or direct base pairing, we found not to be true. And in fact, the data that I just showed you indicates that there is no specific and mappable region, an essential region in nanos RNA required for homotypic assembly in granules, and that therefore homotypic assembly of these transcripts is RNA sequence independent. So again, these results were intriguing to us and suggested that if there should be interactions among these mRNAs in clusters, then they should be non-specific or disordered, and they shouldn't be sequence-driven or ordered. And we wanted to address that in vivo. But first, we wanted to make sure that we can actually detect sequence-specific ordered interactions in vivo using microscopy. And to do that, we turned our attention to OSCAR mRNA. OSCAR RNA localizes to the posterior tip of the developing oocyte. And work from NM Fuzi's lab at EMBL has shown really, really nicely that this RNA localizes to the posterior tip using first active transport, where this RNA finally engages with the kinesin motor to become transported to the posterior pole. But where the second Oscar RNA piggybacks on the first one using a palindromic sequence, you can see here in magenta, where this palindromic sequence is presented within the stem loop that is embedded in the 3' UTR of Oscar RNA. And the Infusis lab has shown this using genetic and molecular techniques and transcompensatory mutations to really show that this RNA is engaged in dimerization events using direct base pairing between two palindromic sequences. And once this Oscar localizes to the posterior pole, it actually also forms clusters there. So we thought that if we use Oscar clusters as the initial readout, and we put a single fluorescent fish probe proximal to this palindromic region, because at the posterior pole, within this region, Oscar will be engaged in direct sequence-specific RNA-RNA interactions and will become spatially constrained. Within that region, this RNA is going to be bound and will therefore form a very small radius of the RNA cluster as reported by this single probe. But if, on the other hand, we now put a single probe somewhere else on the Oscar RNA that is not engaged in sequence-specific trans-RNA-RNA interactions, then within that region, these RNAs will not be spatially constrained, and we will be able to measure a much bigger radius of the mRNA cluster. And this configuration then in vivo will suggest to us that indeed this particular RNA is engaged in specific sequence-specific ordered trans-mRNA-mRNA interactions within the 3' UTR of this transcript. And finally, if regardless of where we put the probe, 5', 3' UTR or middle, we detect more or less the same radius of the RNA cluster, then this configuration is going to indicate to us that this RNA in vivo is engaged in non-specific, disordered trans-RNA-RNA interactions. So to do this, we used storm microscopy, and we collaborated with Eli Rothenberg's lab at NYU and his postdoc Kim Dong, and we labeled an RNA such as nanos with a single fish probe that was conjugated with a photo-switchable dye, such as Alexa 647. We observed very, very long and beautiful photo-switching events, as shown here on this blinking movie. We then reconstructed the image, extracted the data, and feed the data to an autocorrelation function, as you can see here on this graph. And by doing so, we were able to extract the information of the radius of the RNA cluster as reported by that probe. And we first looked at Oscar RNA, and we hybridized probes one by one along the entire length of the Oscar mRNA. And as soon as we hit the palindromic sequence, we recorded a reduction in the cluster radius, about 40% reduction, which was statistically significant, which indicated that this RNA in vivo within the 3' UTR was engaged in sequence-specific ordered trans-RNA-RNA interactions. And this was beautiful because it was aligned very well with the molecular and genetic data reported by Infusis lab. And I would like to point out that this, the radius of the cluster is not indicative of the length of the interaction itself. It just shows where a particular region of the mRNA is most likely to be found. It's a probability measurement. But when we turn our attention to nanos RNA that is clustering in germ granules, we found no such constriction, regardless of where we put the probe. So we really, what this result indicated to us was that in vivo, in germ granules, in these clusters, nanos is engaged in non-specific, disordered trans-RNA-RNA interactions, despite its very high concentration. So the data that I've showed you really suggest the following model, and that is that upon their enrichment to germ granules, mRNAs form homotypic clusters, and that they do so in a sequence-independent manner, but in highly transcript-specific manner, in such a way that these RNAs are able to distinguish between foreign RNA, such as nanos and GCL, and as well as near cognate RNA, such as nanos and its derivatives. We, of course, do not know what generates the specificity for homotypic assembly. But given that small perturbations to the transcripts are enough to cause the mixing of a chimeric mRNA from the endogenous RNA, this result suggests that it's probably the entire property of the mRNP that is really important, not just sequence, not just single structure or a protein bound to it. So in this holistic approach, and this is something we're trying to understand now, we would like to understand how the proteins that are bound to the RNA contribute, proteins that are directly bound, as well as perhaps ribosomes, how the structure is contributing, how the modifications are contributing towards generating the specificity for homotypic assembly. What we also would like to understand is what happens to these mRNAs after they become enriched in these very clustered and highly concentrated condensates. What was fascinating to us was the measurement that outside of the posterior pole, in the somatic regions of the embryo, mRNAs such as nanos have a certain concentration. We measured about 3.2 nanomolar. But once they come to the posterior pole and form these clusters, they become highly, highly concentrated, almost 5,000-fold increase in the concentration within these clusters. So there's plenty of opportunity for the mRNAs to become engaged, possibly into promiscuous and potentially deleterious interactions that would not be beneficial for the embryo. But this is not what we see. Actually, this embryo is perfectly fecund. And we would like to understand how is it possible that in cases such as myotonic dystrophy, when you have these sequence-dependent interactions that form aggregates and cause disease, and on the other hand, we have sequence-independent nanos clusters that are also highly concentrated, but promote fertility. What is the difference between the two of them besides sequence dependence and sequence independence? And how are these potential conflicts that could give rise to disease resolved in Drosophila germ granules and homotypic clusters that form there? And finally, I would like to point out that we see this type of homotypic assembly for every mRNA that we have looked at that is capable of enriching in germ granules. And then importantly, other people have observed such assemblies as well. For example, mRNAs that localize to germ granules in zebrafish, as well as mRNAs that localize to germ, stress granules and processing bodies in U2OS cells. So it seems that this is a common feature of the mRNA that is revealed at sufficient concentration, so possibly when they become enriched in this type of condensates. And we would like to understand why RNAs do this. Is there a function to these homotypic assemblies? We would like to think about these homotypic assemblies as post-transcriptional factories. As I said, that translation, regulation of RNA turnover, prolonged localization, even perhaps buffering of gene expression noise occurs in these granules. And we would like to think that maybe these homotypic clusters have something to do with it. Just as the concentration of this RNA increases once RNA is localized to these granules, so does the concentration of important regulators and factor molecules that these RNAs bring with themselves that could then possibly regulate their post-transcription after the RNA is enriched in granules. So this is something we're pursuing in the lab as well. And in the end, I would like to acknowledge my lab for really being courageous in pursuing these questions. We are at the beginning, we just started our lab, but we are trying our best to tackle these difficult questions. And of course, I would like to thank you, Dewpoint Therapeutics, for inviting us to participate in this seminar and to discuss these discoveries with you. And if you have any questions, I would really like to invite you to ask us and email us, and we're looking forward to interacting with you more. Thank you.
Thanks. It's wonderful. There's a,
thank you.
As you know, there's a approach in Germany. They don't clap. They bang on the table. So we've gotten used to doing that instead of clapping. So thank you.
Thank you.
Questions? I bet there's a bunch of questions.
Hi.
Hello.
Great talk.
Thank you.
I want to ask a technical question about the single molecule RNA fish. So in that slide where you had all the different dots shown and you had like a measure of the number of RNAs per dot, how did you estimate the actual number of RNAs in each spot? Was that just based on intensity?
Yeah, so we are using an algorithm that was developed by Timothée Lyonnais. It's an algorithm that uses the intensity of single mRNA as a proxy, and then we calibrate and normalize all other spots relative to that. Yeah, and so the nice thing about fish, or maybe the bad part too, so it's really great at lower concentrations. We find that about 3.5 nanomolar is really the upper limit. So we can't really quantify anything. We would never be able to quantify beta-actin, for example. But we're very sensitive to lower concentrations, and we are very accurate. But we can calibrate bigger spots nicely, knowing what the intensity of a single one is.
Right. So, and the reason you can't go above 3.5 is just because it gets there's signal everywhere, so you can't distinguish anything?
Yeah, it becomes too dense and you can't resolve them very nicely. So what really helps is structural elimination microscopy. First, it gives a better resolution, and then it gives better signal to noise, so you can differentiate them better.
Yeah.
And we see very nice correlation between single molecule fish data and RNA-seq in terms of RPKM values. So we know that we're doing it well, but it breaks down at higher concentrations. Cool.
Yeah, because one thing we're interested in is doing fish on a bunch of different potentially interesting condensate targets. And one thing I've always thought about is, like, since fish is always a puncta, right? The signal is always a puncta.
Oh.
So how do you decide whether this cluster of puncta is maybe in a condensate or not? So that helps a lot.
Right. So first, this is a real classical problem, right? Because in the end, even if you don't add any probe, you'll still puncta. That's just the nature of biology, and there's background. First, it really helps if you have many mRNA, many probes. So I can send you the protocol, at least for well, I've written a protocol for Drosophila. And then also for yeast, it's more or less the same. And it's always the same problems. It helps to start off with something that you can change the concentration of, so that you can go to a higher or lower regime and you can see how spots change. Because sometimes, we all imagine these beautiful spots, and then you do your experiment and you just don't see that, and you're confused.
Yeah, that'd be great. Thank you.
Right. So I would say, I would start with something that is not as expressed as beta-actin, if that's a mammalian cell. If that's a yeast cell, I would start with, I can give you some examples. And if you're working with Drosophila, I can give you examples as well. Then you would also do, classically, you can do a knockdown to see that the signal disappears. You do a two-color experiment where you mix probes that are red and green. But really, starting off with many probes is better. And choice of fluorophore, in my hands, the fluorophores that we use and are mostly reliable, really nice, they're really nice fluorophores, are Chalcofluorofine 90. We're using Stellaris probes. They're commercially available. They're very easy. When I was working with yeast, it was Site 3 that was really reliable. Far red probes are also okay, but they tend to bleach. And 488 is beautiful too.
Okay, awesome. Thanks so much. Sounds like would be a very good person to talk to if we have technical problems.
Oh yeah. It's not that difficult. It sounds crazy, but it's true. You need to learn to see. It's a little bit at first bias, because as I said, you're hoping to see those published results, and then you look at your gene and it's just not like that, and maybe it's just underexpressed, or maybe it's so highly expressed that everything becomes very homogeneous, so it looks like diffuse background.
Yep, makes sense. Awesome, thank you.
Sure.
Emily, did you have a question?
Yes, I have a question. This is beautiful talk. I mean, I really enjoyed all these diagrams and also the images of all these fluorescent spots. And I used to, in graduate school, I used to do a lot of in situ on the limb regeneration, limb development on the limb beds, where the mouse embryo was taken out, and we do in situ hybridization and see all this morphogenesis environment where all this RNA was during development expressed a different patterning. Also see like the limb bud has the edge AER region lighten up with different mRNA. So it's very exciting to see all this in the embryo.
Yeah.
So I have a question that I think I missed the point where you showed different probe actually had different effect on the same mRNA, where some of them like close to the penta drop or it has more aggregation, but in other region it's a little more diffused.
Right.
I'm wondering what is that?
So we wanted to know, in this, so we observed clustering for all these mRNAs, and so when we did genetic experiments and we were swapping regions, we found that not a single region was required to generate the specificity. In other words, there's no element that would bring two mRNAs together in a specific manner. So that indicated to us that if there are interactions among these mRNAs, and this is a highly concentrated environment, it shouldn't be sequence specific. It should not be something where the mRNAs, even though they're highly concentrated, should be extremely highly packed. So we wanted to address this, and we looked at an RNA where we knew that the RNA was engaged with a particular part of that mRNA in direct base pairing with another RNA originating from the same gene. That was Oscar. So it has this little palindromic sequence that is able to dimerize, and only within that region, that little stem loop. So that means that, so, and then when we looked in the embryo and we looked at what was the radius around that palindromic sequence, because these RNAs in the cluster were engaged in dimerization, they were much more closely aligned to each other than with any other part. Right. Indicating even though there could be interactions among these clustered RNAs in a highly crowded environment, those interactions that are occurring outside of the palindromic region are not, they don't generate as much packing ability as a single sequence-specific region, a palindrome. So it indicated really to us that outside of the palindromic region for Oscar, that mRNA is likely, I'm not going to say flopping around, but the interactions are much more transient label, and that mRNA might be a lot more fluid outside of the palindromic region than within the palindrome, which constrains the mRNA. And then when we looked at RNAs, we didn't see any such constriction. In fact, the radius was more or less the same along the entire RNA as we observed to Oscar outside of palindromic region, which indicated to us that along the entire RNA in the homotypic cluster in granules, we don't observe sequence-specific base pairing. The interactions are therefore disordered, and that the entire content of mRNAs in the cluster is likely diffusible, mobile. There's no spatial, there's no constriction, and that was the relevance.
Great, thank you. I had a really unrelated question. So when RNA was transcribed, it was transcribed as a pre-mRNA where all the intron was also attached, and then it went to processing to cleave off the intron region. I was wondering if the clustering happens when the intron is still attached without cleave it off, because a lot of intron has repetitive sequence where maybe you have your GGG CCC that's kind of more typically aggregate before it was processed, but sometimes if you use your probe only looking at the translated region, you detect it, but you did not detect the intron, which is not cleaved yet. So I was wondering if we know in the germ granule, all this mRNA is already processed, so all the intron was already cleaved off.
So that's a really good question. We haven't thought about that. I'll answer two ways. The general overall RNA-seq data in the embryo suggests that all these RNAs were processed. But only 4% of the RNA actually localizes in these granules of the total, 4% of nano. So it could be that we're not detecting the 4%, or that the 4% that localize have the split. So we don't know. That's a good, we should see. But I can tell you that all the chimeras, besides the last one where it was the full-length nanos mRNA, together with intron, so the entire genetic, the gene region, so all other chimeras actually didn't have introns, and they nevertheless localized and clustered. So I don't think so, but we actually have not verified that this 4% that do localize don't have intronic regions.
I see. Thank you. And I have the last one.
Yeah.
So do you think this is more theoretical? Do you think this is like a storage depot for them, or how they were moving out of it to get translated into functional protein?
So so this is all indirect evidence. We don't actually think that RNAs exit these granules to become translated. And the reason for that is we're developing tools to address exactly that and some other questions regarding translation and homotypic clustering. But nanos RNA has a translational control element that represses this RNA in the somatic regions of the embryo. It binds this particular protein called SMOG. It requires OSCAR, which is almost exclusively found in granules, to relieve the repression of SMOG, imposed by SMOG. So I don't see how it can go in and out so that it would be, enriched, but then if it needs to really translate it, it needs to exit, because it would just be repressed again. And actually it would diffuse away because the embryo is syncytial, and it's about 500 microns across. The volume of germ granules is 0.01% of the entire volume. And I believe that if the RNA cluster would exit, it would just diffuse away. And we don't see any loss of mRNA over time in this embryo. And also, the gradient of nanos protein is originating from the posterior and goes towards the anterior. So we know that it's produced locally, but nobody has really formally tested it. So I think it's really indirect evidence that suggests that translation is happening in granules.
Great.
Thank you.
Alicia, you had a question, I think?
Yes, hi. I was just wondering if you have any structural evidence to support the data that, or the model that these RNAs are disordered.
We don't know anything about the structure of the RNA itself, zero. And we're trying to, we would like to have that data. It's a little bit more complicated just because of the availability of the material. That's number one. 4% of RNA enriches in granules that are only found at the posterior and account for 0.01% of volume. And although these granules have also gel-like properties, they also have liquid-like properties. So purification is going to mess them up a little bit. So, but we would really like to know what is the structure of the RNA itself in granules. We don't know that. All we know is that the interactions among them are not anything that is anything that's ordered or sequence specific, and that's what we're calling disordered.
Is there a way that you could do NMR or any other method in order to show that?
So our best was to, so the genetic evidence, when we were pairing mRNAs among that shared regions, And we saw that, chimera and the endogenous mRNA never came together, even though they shared exact regions. This chimera recapitulated all the essential features of the endogenous RNA. So we thought at one point, if we add all the mRNA sequences back to the chimera, we should recapitulate the entire RNA, and they should come together if a single stretch or combination of these stretches is required to generate specificity, and we didn't see that. So that's why we're thinking it's the entire mRNP and all the characteristics of that mRNP that are important. And then with STORM, we saw the same thing. Now, we don't see any particular constriction. We are not saying that there's no interactions. We just say that there's nothing that is sequence-based, there's specific, right? And that's why we're saying that these interactions, should they occur, are disordered. They're not sequence-specific ordered.
Mark, I have a question.
Mm-hmm.
Great.
I'm just searching for an analogy here.
Oh.
In
The
music,
same thing that was happening to you yesterday, Anne. You keeps muting you.
yeah. So in music, you can listen to a piece of music, and it has a feel, right? This is a Baroque piece. This is Beethoven. This is Schubert. This is something entirely different. Aaron Copland, right? If you're really familiar with music, you can just listen to a little excerpt. And my daughter and I would play this game of guessing the composer or guessing the piece.
Mm-hmm.
But the notes are all the same,
Mm-hmm.
right? And yet there's this feel that is different, right? And somehow I'm wondering if, in a way, the cell is able to listen to music. And this mRNA has a different sound, the whole, right? Even though the notes, the nucleotides are all the same, it's not that. It's something
Right.
bigger. Does that make sense?
Right. No, we, especially when the paper was in review, we're really trying to, because there's a little bit, it is a bit confusing, right? We haven't shown what is required. We just shown that something is not, and this is primary sequence. And that primary sequence, either by forming a secondary structure that would be conserved. We are not saying that it's not important. We're just saying as soon as you modify this RNA a bit, and we modified it either by inserting an eGFP open reading frame in one, so that with the entire length of the mRNA, with the introns, the promoter and everything. So either we inserted the eGFP or we inserted, it became different.
Right.
Even though it is still the same mRNA, it recapitulated all other features that we could characterize for that mRNA: translational onsets, translational repression, localization, clustering, and yet it was behaving differently.
Yeah.
That's why we're saying it's not as simple as one defining or mappable structure. And on the other hand, I think that what makes it, so what, in terms of your analogy in music, you can listen to a little piece and say, oh, that's Coltrane. And in our case, you look at the 3' UTR and say, oh, that needs to be bound to germ granules. But once it gets there, there's all this other stuff that makes this RNA nevertheless behave differently. So there's little nuances that then matter. And what I think is really interesting and important, it's because of this high specificity, perhaps, that you don't really see often clustering outside, even though these RNAs share features with many, many, many other RNAs that should, in principle, make them come together. And I will talk about exactly that. This is all very preliminary. We as I said, I just started, but we do have sequences in these RNAs, especially in the 3' UTR of these transcripts, that make them specifically bind to each other. And yet,
yeah, I mean, you have that palindrome interaction. So that can override everything. But I guess what I would say is that if you have a piece that's Schubert overall, and then you stick in a little piece of Mozart somewhere in there, it doesn't change the Schubert overall. So that's when you did this, that's what you're doing. And what we just don't have a way, but the cell must have a way of sampling. This is, you know, EGFR, this is not.
Yeah.
And it's not any one small little, you know, piece of the RNA. And so we're reaching for a different metaphor, but are reaching for a different measurement. It's kind of like being colorblind and also now being able to see, oh my God, this is green, and we're only looking at black and white. But black and white, just looking at the tiny, tiny motifs in the RNA,
Right.
and it's clearly not that. It's
Right. Right. We call it solubility. I mean, I know it's common in the phase separation, but if they come together, they're soluble. If they don't, they're not. And what makes them soluble or not soluble must be some property of the entire molecule.
Yeah, but to me, that's a very pragmatic description, but I think it doesn't get to I think it's probably more poetic than that when you actually, as you do more experiments, it's gonna be something bigger than that. It's super cool, beautiful, beautiful talk. Thank you.
Thank you so much.
Yeah, it really was. Obviously, we could keep asking questions for another hour, but we're out of time. But maybe if it's okay with you, if we have other questions, we could just follow up with you.
Sure. Yeah. And also we love to hear your perspective on this. You know, as I said, we're a young lab. We like questions. We're trying to see how other people think about our problem, and sometimes we're a little bit too mired in what we do. And we certainly, if you have questions, just email the question to us. We're excited that we can participate. Thank you so much.
Oh, that's great. Yeah. So thanks again, and thanks
you
for everybody on the call as well for joining and for the great questions. So
thank
all
you.
right, everybody have a great day.
Much.
Thank you. Thank you so much.
Bye.
Yes, thank
Okay.
you. Really awesome.
Everybody.
Bye-bye.
Thank
Bye-bye.
you. Bye.
I guess somebody has to stop the recording.